I'm Charlie Wykoff, retina specialist in Houston, Texas. Absolute privilege to be moderating this session with Arsham, Sheybani over here on the left side, right side if you're out in the audience, from the University of Washington in St. Louis. And I appreciate you all staying. It's been great energy today, and we're closing out a phenomenal day, hopefully saving the best for last, although we will have to see, as there have been many great talks and great energy. The goal of this session is to put regulatory issues front and center. There have been some slightly controversial, at least among the retina specialists, draft guidance in the retina sort of realm, and similarly some challenging things to digest in the front of the eye and more broadly across eye care as well. So we're going to take a deep dive into that. As you can see, we've titled this 10 Perspectives to Navigating Today's Regulatory Landscape. So with that, I'd love to give all of our panelists and awesome regulators a moment to introduce themselves. We'll start here on my side and go down the row, and then we'll jump right in.
Yeah, Steve Schallhorn, Chief Medical Officer for Zeiss Meditech.
Dave Boyer, retinal specialist in Los Angeles, California.
Wiley Chambers, Director of the Division of Ophthalmology, Center for Drug Evaluation and Research.
Malvina, Eydelman, Director of Office of Health Technology 1.
Gary Novack, PharmaLogic, and UC Davis.
Eris Jordan, Global Vice President of Clinical and Medical Affairs, Aurion Biotech.
Hi, I'm Chuck Hess. I'm Vice President of Commercial Development for Bausch and Lomb.
I'm Gus Gazzard, Director of Surgery at Moorfields Eye Hospital, and I think token Brit.
So the session is going to be a little different. We're going to present slides. We're actually encouraging discussion amongst the panel. The slides are going to be kind of launch points for our panelists to pose some of these questions to our FDA representatives. And we have a very diverse group of folks. So I think we'll start with our first topic here.
I think Eris Jordan, this one might be for you.
Sure. Primarily, thank you for the opportunity for being on the panel with all of you. I realize that it states that this is a retina main panel, so I'll go ahead and kick it off with an anterior segment question. So Aurion Biotech, as most of you know, is working on a novel cell therapy for corneal endothelial disease, and primarily, not only are we novel and first-in-class treatment, but we're also a combination product with cultured corneal endothelial cells falling under the CBER jurisdiction, in addition to a Rho kinase inhibitor, small molecule, falling more on the CDER side of the agency. So communication with the agency really does become a paramount as it informs our development pathway. So we did notice that a guidance just recently came out from the agency in September regarding communication with sponsors and the agency. So we appreciate the agency for I know there's, I think this is a different question that you wanted me to ask,
Yeah.
but a little bit of a change in plans here.
Go for it.
So going a bit out of order. That's right. Yeah, so this is question number one was
Yeah.
That's fine. Ask the question that you're posing.
last minute change, but I'll go ahead and ask my first question nonetheless. So regarding the guidance with communicating with agency and the sponsors, I wanted to commend the agency for providing the topics for us to discuss. Is there anything that you can do to highlight the main topics that the changes to the draft guidance for communicating with a sponsor?
Depending on the center that you're talking with, I'm just going to speak for the Center for Drug Evaluation and Research. We assign a project manager to every application that comes in, and that project manager is responsible for navigating the FDA component to figure out who it is that can answer your particular questions. In addition, we don't turn down meetings, period. So if you have a question, you ask for a meeting and we will talk to you and answer your questions.
So I believe you were talking about the pre-subs?
That's correct.
Okay. So for those of you in the audience, the slide projected and the questions being discussed is different. That's hence the confusion. So as far as pre-subs, there is a number of different types of meetings that CDER has, and I think that was the question, as far as A, B, C, D, and E. So as for the Center for Devices and Radiological Health, there's basically one main type of a pre-sub meeting, and we always get to meet and discuss and provide written feedback within 70 days of the submission. So the sponsor gets to choose whether they want just a written feedback or whether they want to meet with us in person as well. And unlike CDER, there is no limit on the number of questions you can ask when you submit a pre-submission to the CDRH. Having said that, you know, it's always helpful for the sponsor to stop and think about what are the important questions. Having 40 questions that have no consequence on the design is usually not a very good way to proceed.
I'm going to move us forward just so we get time to go through all of these. Thank you for those great insights. So, yeah, where are we?
Was there a change last
No,
why
they
did we
did
miss
change it.
that?
There you go.
Are we got the right slide deck up here? But you know, okay. So, Dr. Chambers, I'm gonna ask you a question. Right, this concept of non-inferiority versus superiority design comes up all the time. And I guess the key concern for us, looking at sort of the clinical practice of retina, is if there's no biological rationale for why a new drug, a new product should be superior, if the goal is for it to be non-inferior to drugs on the market, how can we work together to design trials that are demonstrating non-inferiority for something that is developed to be a more durable agent, but has no rationale to be superior without undertreating patients in clinical trials?
So you cannot do a non-inferiority margin, or you cannot do a non-inferiority trial if you don't have a non-inferiority margin. So there's no way to go and make that comparison. So we don't allow non-inferiority trials when there is no non-inferiority margin. That said, if you think your product is superior because you think it has a longer duration, then you demonstrate that it really is superior with that longer duration. If you think it is the same and there is a non-inferiority margin for things like AMD, then you test it against the product that has the non-inferiority margin with the same dosing regimen. But if you're going to claim that you can go out longer, then you need to prove that your product goes out longer and the comparator doesn't. That's superiority.
And one follow-up, thank you for that clarification, Dr. Chambers, very, very helpful. How do we do that without underdosing patients? Just for example, we know that aflibercept, you know, needs to be dosed regularly, at least with all of the trials that have been regulatory-driven trials. And if a drug is being developed that is meant to be a one-year trial, should we dose patients once with Eylea and then just wait till they lose vision? That doesn't seem like it's a good patient-centric approach.
With all due respect, the retina community does not currently follow the labeling of products.
Fair point. Point taken.
So to say that there's a problem with not following the labeling.
Good point. Understood. Thank you. I rest my case. I think the slides are a little bit out of order here.
They're definitely out of order.
I think, Gary, you had a question that was next.
Right. So we have it as 3. It might be 1 on the slide. So there seems to be a growing need to work with more than just one center at FDA. It might be due to this genus decision. I wouldn't have enough time to explain what that is, or real combination products that are a device and or a drug and or cell or gene therapy. And so simplistic things like, I don't even know where to put what in certain applications. More complex, I'm going to quote a CFR, 20 CFR 820 about bottles and devices, and also how do we keep in common different standards for efficacy between the centers? How can we best work with different centers?
Okay. I know this created a lot of confusion, but if you give me about 5 minutes, I think I can explain it. But you have to be patient.
Go for it.
Okay, so the genus decision, a very simplistic view, is to say that it said that no longer can the devices be regulated as drugs. So, but for those of you who don't know what a combination product is, it's an entity that is comprised of 2 or 3 different types. So it's a drug, device, and biologic. Now, because of that, there is an Office of Combination Products that determines which center, how is that product gets regulated. And it has to do with a primary mode of action. So for every combination product, one of those components is primary. And if you don't know which one it is, then there's something called RFD, or request for designation, and the Office of Combination Products can tell you. Once the primary mode of action is determined, that's the center that is the lead for your application. Going back to the genus, that's how all the confusion started. A couple of years ago, before 2021, the dispensers that were packaging together with the drops were considered drugs. And then after the genus decision, then that is now considered a combination product. So to clarify that, there was a guidance that was issued that delineates, it was in 2022, it actually steps you through what to do with any combination product. There's also a separate guidance specific for ophthalmic dispensers that says that it is now going to be a combination product. My group had a device panel meeting about a year ago in order to try to classify them, and now we're going through the classification process. But irrespective of that, back to your original question, which is how do I make sense out of all of this? The first thing you do is you figure out, A, if it's a combination product, B, you have to decide what is the primary mode of action. Then you need to ask for a pre-meeting, and in your pre-meeting, you need to ask very specific questions for each center. Because what happens is, let's say CEDAR is a lead, but if there's a device component, in your pre-submission, what you need to do is say, can we please get following clarification about the device component? And you go A, B, C, D, and then the consult comes to my group, and my group provides specific recommendations. This way, when your marketing application comes in, you're not shocked. Because eventually the marketing application goes to every center that has a part of the combination product. This is Combination Products 101 in 5 minutes.
But the ophthalmic dispensers don't go to your group. They go to one of the other offices. They go to 3.
It doesn't matter.
They don't go to
Still,
one.
it doesn't matter. It's still devices, and now they're going to be. Now we're going through it.
And they have been inconsistent.
Well, and now this is sort of irrelevant to this
From
discussion.
product to product. Basically, we had a system that worked, and it was thrown into disarray because of the genus decision. Congress altered all parts of the genus decision except the ophthalmic portion.
So I disagree. I think the system is going to be much smoother now. We have a new leadership in the Office of Combination Product. We're going to be getting a lot of good clarity. It's actually somebody who I hired a few years ago. So he is very good, and he's leading the efforts now. So I'm very optimistic.
Well, thank you for that clarification for all of us. Dr. Boyer, did you have a question about this?
I have a very quick question. I don't know who to address this to. Why does it take so long to get a pre-filled syringe? Where does it go? Does it go to you? I mean, it's taken companies years to get a pre-filled syringe when you know there's safety involved.
I'll let Wiley answer that.
I didn't know who to ask.
because it is not with your group. It is with a separate office who sometimes asks lots of questions, resulting in a 102-page review in one case for a needle. I don't think it should take that long. The holdup is generally with the chemistry reviewers in the biologic group, but they like to ask lots of questions.
Well, thank you.
Dr. Boyer, will you please ask your question? Yeah.
Yeah, I think that many pivotal trials for approved are having to do double masking, and they're using sham intravitreal injections. You're an ophthalmologist. You know that a sham injection of even balanced salt solution carries with it the risk of floaters, PVD, retinal tear, and infection. The articles that came out from the DRCR net were sort of mixed. Obviously, if you've had an injection of a drug in one eye, you'll know if you're getting it in the other. But now we're doing suprachoroidal, so nobody knows what to expect with that. You're also doing drugs that don't really go through the vitreous. And Hawkins had two papers that really asked, do you really even need to do this sham injection? Because they didn't find any difference when they did an open label study versus a controlled study where sham injections weren't given. There was no difference in the eventual visual result, which is obviously what we want. We have our photographers and we have our vision takers being masked. Do you think that we're really gaining, or there's a risk to the patient that's difficult for me as a clinician to accept?
So the only time to use sham is in an adequate and well-controlled trial. Let me back up. Adequate and well-controlled trials, by definition, you need to minimize bias to the extent that you can. Sham inherently doesn't minimize bias. Patients do frequently know, I take issue at the articles that you're referring to, the data doesn't actually match what the conclusions were. Patients know, we have lots of experience with patients knowing when they receive sham versus when they receive an actual injection. To the extent suprachoroidal injections occur, you're not entering into the vitreous, you don't have the floaters, you don't have any of the rest of the things. I don't think it's the same issue. To say that a saline injection is riskier than a test product, which has only been used in a few patients, also doesn't stand up. The saline injections have been given for a long period of time. We know that they don't do harm except in very rare conditions, and that same risk applies to the initial product. So you're really comparing apples and orange, comparing apples to apples, not apples and oranges. So because you don't need to do shams, we're going to continue not to recommend shams.
I mean, the only question becomes, I mean, Hawkins pretty much showed that there was no difference between the group that had open label and the group that was in a controlled trial. Because vision's really what we're after. I'm not sure which group tries harder, the group that's getting the injection and the group that's not getting the injection. So I have a disconnect in that regard.
I can tell you open label trials historically always look better because patients try harder.
So when we're thinking about how some products are approved based on structural endpoints and some on functional endpoints, you know we're now in a landscape where we're looking at alternative endpoints that are kind of more meaningful for quality of life. Do you have advice or guidance for firms that are looking for approval? How do they meet FDA standards for quality of life, especially when sometimes there's not great metrics like in glaucoma-specific quality of life measures?
I'll take this one.
Okay.
So I'm sure many of you know that I've been extremely passionate about incorporating patient voice in approval of new technology. And there are many different ways to do it in a scientific manner. While I have all of your attention, I want to invite you to a session that we're putting together this Saturday together with NEI and AAO on the patient voice in ophthalmology. The key message is that there is a way to do it, that you need to be very systematic, and you need to collaborate in order to achieve it in a timely, economic manner. But there are quite a number of PROs that I've been fortunate to lead that are now finalized and are available and are free. And just 3 weeks ago, we had a workshop together with NEI where under Michael Chang's leadership, we discussed how to go from having PROs and spending 10 to 12 years per PRO to, for a particular condition, is there a way to design the future of PROs so that we can create, as we call it, the super PRO, something where we can utilize it across all of the ophthalmic care. And we have fantastic new approaches to discuss on Saturday. I'll keep you.
I think we have.
Steve, Dr. Schallhorn, did you have a question to ask the regulators?
Yeah, I've got a question just on the same theme of patient-reported outcomes. There's obviously a lot more emphasis on it. There's a lot more interest in it. And from a clinical perspective, aside from safety, it really represents the most important clinical outcome, right? How, in particular, how satisfied the patient is with the eye care provided, the procedure outcome, et cetera. And you've done AAO, jointly with the AAO and industry, developed these pros, as you mentioned before. So the question is, let's just take devices and just take IOLs as an example. In clinical trials, they've been the secondary endpoint. So do you foresee where a patient-reported outcome will be a primary endpoint in any of these studies starting up? And I guess a second question is sort of related to that, and that has to do with in a new, say, IOL design, do you see the patient-reported outcomes, there's a metric against, where your patient-reported outcome metric against, say, a presbyopia correcting versus monofocal, as you do use that metric for other outcome metrics, defocus or visual acuity. Do you think PROs are going to be bumped up in that regard, primary outcome, and used for other measures like that?
Yes, but not for every condition. So last October, we had a workshop in collaboration with Dr. Sahel's group at University of Pittsburgh for bioelectronic implants. And those, as you know, for people with profound visual loss. So in that population, we actually had a probably 4-hour discussion of what is the most appropriate outcome to assess for efficacy in clinical trials of bioelectronic implants. We have an old guidance that's outdated that talks about utility of functional assessments, mobility, et cetera, as the essential outcome in addition to visual acuity. But where is the line? It's shifting. And I think the more, and that's why I would love for everybody to come to our session, because I truly believe that where we're going depends how comfortable all of us get with using PROs and how easy it is to use them across all of the conditions. So yes, maybe for some.
Yeah, yes.
Thank you. A comment rather than a question. Speaking as somebody who spent greater than 8 years of his life leading a multi-center randomized controlled trial where we had a mandated patient-reported outcome measure as the primary outcome, which was negative, and 3 other patient-reported outcome measures which also failed to show a difference between the 2 treatment arms, but nonetheless were sensitive to the decline in with age. And yet that same trial still has had some impact on practice in some areas. I really think we've got to put a lot more effort into showing that any of these PROs are both sensitive enough and meaningful, that they have a valid and meaningful outcome for the patients, because we had that mandated by regulatory authorities in the UK for a non-industry trial. It was an NIHR HTA, Health Technology Assessment funded trial. But that was a painful lesson there, and has led to the NHR moving away from PROs as primary outcomes. I really think we need to be careful about mandating those as primary outcomes.
So first of all, nobody is mandating it. I think it's always going to be an option in the future, potentially. And you need to think about the disease and whether we're talking about using PRO's efficacy or as safety. Because very often, right now, we're using it as a part of our safety assessment, especially visual symptoms. There's no better way to assess visual symptoms that patients get from either LASIK or AIOLs than a PRO. So, agree to disagree.
I'm going to bring us back into retina for a second. Dr. Chambers, fortunately, we've been able to develop anatomic structural endpoints with your assistance over the years, geographic atrophy, VMT, diabetic retinopathy, et cetera. What would it take to develop an anatomic endpoint for wet AMD, whereas vision said and done, full stop? Because as you know, all of us in the real world, in the US and abroad, use anatomy, use imaging and exam to guide treatment initiation and treatment continuation and intervals. Is there any data that you could see for fluctuation data or absolute amount of fluid that would change the opinions of the regulators on allowing an anatomic endpoint for approvability for wet AMD?
So there are anatomic parameters that are used as the primary endpoint to establish efficacy for products currently. Absence of photoreceptors is used as either the rate of loss, you slow the rate of loss of photoreceptors, or there are other anatomic preventing retinal detachments. So we do have measures that will take the anatomical finding if we're clear that that is of benefit to the patient. I don't think you're going to find anybody in the room that's going to say not having a retinal detachment is not a good thing. It's also, every patient will tell you, was not a good thing to not have a retinal detachment. There are not that many characteristics where it's very clear-cut that the anatomic feature alone is of benefit to the patient, as opposed to actually measuring visual function for the patient. I mean, we have a great availability of measuring a wide variety of different visual functions that patients need to be able to do activities of daily living. And that's what we've been focusing on for endpoints.
Let me just jump in here. I want to make sure folks understand that the regulatory requirements aren't something that comes out of thin air. It's based on scientific evidence. And so if there is a significant consensus on the scientific evidence, then we will accept it. And the American Academy of Ophthalmology is truly unique. All of us are very lucky to have such a strong professional organization. The consensus statements that Academy publishes are very often used by my staff as a guide to figure out what is acceptable and what is not. So basically, back to your point, it's not AMD, it's anything. If there is a consensus by a large group, such a professional organization, that this is indeed a good indicator, then probability of regulators accepting it is very high.
I just have one follow-up question to that. And I appreciate greatly, and it gives me more insight as to what's going on, but we're not talking about validation at this point. What would it take to get an endpoint such as a maze or a loss of the thickness of Brooks membrane? What would it take to become validated? In other words, if all the groups, the Macular Society, Retina Society, all the groups got together and said, we think that this is an endpoint that's valid, is that something that you would take, or do we need to do many studies, and how big the studies would have to be to?
Well, it depends on what the consensus says. If the consensus says that there is, this is what's needed and this is how it needs to be done, then we'll probably accept it.
Thank you.
There are differences in the way the law is written between drugs and devices. That is not true for drugs. the data to go and support an endpoint needs to be available to be able to show that the benefits to a patient outweigh the risks. A picture on a OCT is not necessarily valid to a patient as being improvement. If you go and thin the retina, in many cases you call that macular degeneration. You go and make it thicker, you call it macular edema. Just moving the thickness, changing the thickness of the macula is not going to be considered as being a primary endpoint.
Okay, Gus.
Thank you.
So the next topic, for your opinion, was going back to a term coined in the 70s, that the drug lag, that the introduction of medications in the 70s was perceived as being delayed in the US relative to new drug introduction in Europe, and that has waxed and waned over the intervening period, clearly long predating my personal experience of it. But what do you think is happening now? What's your opinion on how it is now? Because there are firms that have been selecting countries outside of the US for initial clinical studies and clinical evaluations. Clearly, that's going to be a balance between the risk to the patients and protection of patients from harm, from potential harm from novel devices or novel drugs, and yet at the same time there's a risk of delaying innovation and delaying introduction of those same innovations.
I can start. So that definitely was the case a couple of decades ago. I think especially in the last decade, at least at the Center for Devices, I'll let Wiley talk for drugs. At the Center of Devices, we have put the North Star. Our North Star is bringing safe and effective, innovative devices to US patients first in the world. And due to that, we have introduced quite a long number of programs that help to expedite innovation. I don't want to take too much time, but we have Breakthrough. We have something called STEP, which I don't know if many of you know, but Breakthrough, just like Breakthrough, it's for devices that offer additional efficacy for serious conditions. STEP is something we just introduced within the last year, which is the same thing for for technologies for either diagnostic or therapeutics that are safer. So for things like that, we actually have expedited processes. I personally get involved in all three divisions, Breakthrough and STEPs. We try to, every manager gets involved. We have expedited meetings. We have ways to expedite preclinical, clinical, and also reassess pre to post market shift as to where we draw the line. There's HDEs, there's many different programs. But, you know, no matter what FDA does, we cannot do this alone. In order to bring innovation to US first in the world, everybody in this room, everybody in the ophthalmic community needs to work together. Everybody in the medical device ecosystem needs to be on the same page. And going back to endpoints, that has been the topic of conversation for a while, we cannot bring innovative technology to US if we don't know how to evaluate it. And trying to decide and establish endpoints for innovative technologies is something that needs to be done in collaboration of all stakeholders. And again, I want to give kudos to AAO. They have been an incredible partner for us. We have held numerous workshops in many different organizations, including AAO. As an example, you know, when we had a workshop on AIOLs, I think it was 2014, there was no even term of EDF, extended depth of focus. And here we are. We figured out what the term is. We figured out, we established a task force. We worked long hours together with both academician and AAO and industry to figure out how to evaluate these innovative technologies. And now we all can benefit from the fruits of it. So, Wiley?
On the drug side, the statement that you're referring to has not existed in the ophthalmology space for ophthalmic drugs, certainly not in the past 35 years, with the exception of combination products where each of the two individual ingredients were already approved. And Europe will allow combinations to be put together, not because each one does something, but because it costs less. And that's not a criteria that's used within the United States.
And compliance.
I'm sorry.
And patient compliance. And cost is not the only criteria.
We could have a further discussion about that. I don't think that's true. But as far as studying products, with the exception of a period of time when there was a PharmTox team leader who provided requirements that were beyond what had been asked for in the past. First in human was generally in the United States for ophthalmic products. And that has now returned to the case, with the exception of sometimes it's cheaper to do a product outside the United States. It's not a regulatory mechanism, it's cheaper.
Chuck, do you want to jump in?
Yeah, that's great. Thank you. And we all understand, in order to gain approvals for, a lot of these technologies, these drugs, these devices, a product must either tie, to an approved therapy, which would typically be considered equivalence or non-inferior, or actually beat a negative control or an approved therapy. So that would be more superiority. And I just wanted to ask, how does FDA view these? Are they both still very useful in evaluating technologies? Are they acceptable to FDA?
So I'll go first. Okay. So, devices are better than drugs for many reasons, but devices are different than drugs in that we have 3 different classes, and depending on the class, what you need to demonstrate to get on the market is different. So in that, it has to be a premise. So for Class 2, you just have to demonstrate substantial equivalent to a predicate, which is another legally marketed device. Now, for PMA or for Class 3, it's just demonstrating safety and efficacy. So in that the regulation already drives what kind of trial we're talking about. But as far as SAP or statistical plan, we always say come in and see what it is and propose. We never really say it's 10%, 15%, because every clinical trial is just a little bit different in that we have so many devices, and the risk benefit is what we're trying to get at, and we're always trying to figure out what is the balance of that risk benefit that will get us the comfort level. So there is no magic number.
So on the drug side, does not operate the same way. You need to do adequate and well-controlled trials. Non-inferiority margin is an approximation of what a placebo would do if a placebo was in the trial, but you don't want to include a placebo. So there is methodology to determine what you believe a placebo would do in a trial that doesn't have a placebo. It's based on having, with some other product, a placebo. So if you don't have placebo-controlled trials, or the trials that were placebo-controlled are inconsistent with one another, or there are not enough of them to be able to do an approximation, you cannot derive a non-inferiority margin. So currently, with the products that have been approved with geographic atrophy, if you will take a look at the trials side by side in each of the 2 cases, they are not exactly the same. there is insufficient consistency between the trials to derive a non-inferiority margin. So there is no current non-inferiority margin for products for geographic atrophy with the approved products. Will that occur sometime in the future? Will additional trials be done so that we can derive a non-inferiority margin? I don't know. But based on the currently available data, it's not possible to do a non-inferiority margin. And that's why you've heard the comments that I've said earlier, that you would have to do a superiority trial against something. That something could be an approved product, it could be a vehicle, it could be a different concentration of whatever the test product is. There are lots of different options of what it could be a superiority trial to.
Dr. Chambers, can I follow that up? Thank you. That's super clear and very helpful. If it's an anatomic endpoint and not a subjective endpoint, for example, imaging, is it still required to do a superiority trial against a product that some physicians may think is not safe, or against BSS, which has risk? Is there any way to do that in a sham-controlled injection setting?
There is not a reason to be doing a sham-controlled trial when you have other options and still be considered to be an adequate and well-controlled trial. And you would certainly, with virtually all of these drug products, have the opportunity to do a different concentration and show superiority of one concentration over another. That would be a superiority trial. That would not have any of the risks that you're talking about. It would, in fact, potentially give patients an opportunity for benefit, and you don't actually know which is the better group when you first start. So that's equipoise.
So in a situation like that, that's very helpful also, thank you. Could we have a sham arm and then 2 doses of the study project and achieve superiority to the sham, but not to the second dose? Would that be a potential positive outcome?
There is no reason to include a sham.
Thought I would try one more
You
time.
don't need it. It would not be a benefit in this particular trial in this case.
Thank you. Gary, do you want to finish up with the second to last question?
Right. Can I ask the CBER question?
Of course, yeah.
So, well, I don't know which number. We've
Yeah,
had
yeah,
a
we'll go back.
little bit of confusion in the slides. So, a lot of interest in the room here, of course, in cell and gene therapies, for whom I'm pretty sure the primary division center is CBER. And at least at the time I wrote the slides, I didn't think we had an ophthalmologist in CBER, and I didn't think the preclinical people there had a lot of ophthalmic experience. Can you give us an update?
Sure. There are 3 ophthalmologists in CBER. We actually 2 have joined not too long ago, and I made sure that they're welcomed by my very large team of ophthalmologists, and they have, we did the meet and greet, and so they will be educated and ready to go. CBER does not have currently vision scientists, so they do consult my group for vision science.
There is a very close working relationship between the 3 ophthalmologists and myself and the rest of our team. We meet with them, if not every 3rd day, at least once a week for the 5 months that one has been here, for the 3 months that the second has been here, and for the 1 month that the 3rd ophthalmologist had returned to CBER. She had been with CBER before.
But the good news is it's coming.
That they know how many eyes the average human has, right? That's a good question. Should I try the other one really briefly? Okay.
Sure, go for it.
Eyes versus patients. We know it's a 50-year-old discussion how you do that strategy. And without reading all the details, is the criteria different for a local therapy versus a systemic therapy for efficacy? I tried to do it succinctly.
You can start for a change.
In general, we prefer products that are going to be used once they're marketed in both eyes to be studied in both eyes. Typically, you have the choice of whether you decide to average the eyes, take the worst eye, do something, but because there's potential correlations between the eyes, we generally don't do both eyes independently. We generally don't do contralateral eye comparisons for a wide variety of reasons, including that it's not consistency between the two eyes. So most of the time we'll do our analysis by patient, but there are some conditions where we do it each. We think there's the sufficient independence that we can basically get 2 responses out of each eye, and when that's the case, we do it.
So for devices, if there is a device that is going to be used monocularly, and if you can truly demonstrate that its use will not have an impact on a contralateral eye in any way, then you can use eyes and not patients. That's as succinct as I can get.
Appreciate that. Or can I try it again? And sorry, it's a drug question. Oral versus local, is it still the same standard of efficacy? Sorry, I think you've talked about this previously, Dr. Chambers.
Yeah, in diabetic retinopathy, an oral product is going to affect both eyes. So we want an endpoint that uses both eyes. For a local therapy, including intraocular, just because there is some systemic absorption, we don't think that's where the main effect is, and we'll take endpoints that are for a single eye.
All right, to round things out, be a little bit more specific for devices and kind of clarification for what the FDA, what their role really is. Is it about making sure that a device is safe and does what it claims to do? Because right now I think there is a conception, whether it's true or false, that it's involved really in comparisons and dictating where the market goes. If you have a surgical procedure that might be fine-tuned for 30 to 50 years versus something novel that has not had time to hit the market and be fine-tuned, is that fair to compare the two? Or should we be focusing more on efficacy of what they claim and safety?
So again, depends what kind of a device you're talking about, whether it's a class 2 or class 3. Or class 1, which you might not need to do a trial altogether. So if it's a class 2, you just need to demonstrate, as I said before, substantial equivalence. For class 3, they stand on its own, so it's demonstrating safety and efficacy. So you might not need to compare it altogether. There are certain ones that we can potentially accept. I mean, take IOLs. You know, if for IOLs we have ISO standards that delineate adequate endpoints, so you don't necessarily even need to have a comparator arm, which is another difference between devices and drugs.
Thank you. With that, I would like to close the session and please help me thank our panelists, in particular Dr. Chambers and Dr. Eydelman, for being the target of many, many questions.